As as Sarah mentioned, my name is Craig Macintosh. So today I'm going to give a talk about a type of genetic therapy that we're going to try to increase the amount of protein that the DYRK1A gene makes. I was hoping to have a little bit more data, but we've had a problem with our university moving over to a new system, which has delayed all our grants. So it's been very frustrating. All right, there we go. So I was just going to give a brief background on antisense oligonucleotides. They're called ASOs for short. They're basically a synthetic form of a DNA-like molecule, and they basically bind onto your genetic information with really a great amount of selectivity and a great amount of specificity. So if you sort of think about it like copying a few words in a book that are continuous, the more words you string together, the more likely you are to probably only find one sentence with those exact makeup of words. So in this case, we design the compounds to target a specific area of a gene message. Most people are familiar with a type of mechanism called RNase H, which has modifications on it that allow it to reduce gene expression and therefore reducing the protein that the gene makes. But obviously in this case, this mechanism is not suitable. So there's a lot of different molecules that we can use. Basically, we can't use a molecule that's unmodified. So we fully need to modify the molecule in order for it to enact its mechanism that we want. So fully modified ASOs have a lot of mechanisms that they can use. So for those of you that this might be too a bit confusing, basically what it is we're either trying to remove or include a bit of a gene message, just like trying to include a few extra words in a sentence or in a paragraph to beef it up in terms of the information, or if we want to cut it down to reduce that information or reduce mistakes that might be in that message. We can also remove sections that cause a similar mechanism to the gene knockdown, or we can actually use it now in sort of new mechanisms that are coming to light. An old mechanism was, if you think about trying to turn down a gene message, we basically stopped the production of it, where the mechanism that I'm trying to use in our lab is basically by hiding negative elements that decrease the amount of expression. So the way I like to explain it is, if you think of Goldilocks and the three bears, a lot of gene messages have potential places where they might start. So in this instance, that would represent the 3 bowls of porridge. We, like Goldilocks, didn't know which porridge was the right temperature, and the cells don't really know that either. So they'll start to work off a bowl that might be too hot or too cold at one time, and then only find the one that's just right. So what we're trying to do is actually just eliminate the other 2 bowls, so that the only option for Goldilocks to basically take is the bowl of porridge that we want, therefore reducing the time that that decision is made, so then therefore more protein is made. And that should hopefully overcome the genetic mutations that's associated with DYRK1A syndrome. This is a fairly new mechanism. AO's been around for decades, and this was generally first reported in 2016 and then further developed on from there. It's been shown to be effective both in cells, so in the lab, in various types of tissue, so in muscle, in skin, and various other tissue, and it's also been shown to work in animal models as well. So the proof of concept that these can be used effectively to increase protein expression is there. It's just about finding whether we're able to hide the right bowls so that Goldilocks really does find that forage in the most efficient manner. So as I said, coming back to the Goldilocks analogy, this gene has a lot of different start sites. So it can start at various regions of the gene, but generally there's only one active start site for the protein that we want. So what we do is we're going to aim to sterically block them, which that just means basically physical blocking. So we'll physically block or hide those start sites with various types of chemistries and hopefully increase the protein expression. This is just a genetic map showing you how the different start sites, it's like a book, it gets read left to right. So you can see that these all start in different regions, while these ones all start up here. So when you're trying to screen a drug that's based on the position of a start site, the more start sites that you have, the more complicated it can be. So what makes a good start site? These are all really technical words, but really the Kozak sequence is just an area around a start site that the genetic machinery uses to recognize that. So if it's a strong sequence and you cover it, or it's present in that gene, there's less gene made. The length away from the normal start site of the gene, and various other factors that can get influenced, including sort of the structure of the compound in 3D. So what we needed to do is we needed to determine which isoforms or which specific versions of the gene are present both in patient cells as well as a neuronal cell, because obviously most of the phenotype is thought to occur at a neuronal level as well as obviously other levels. But a therapeutic is, most likely to be administered in the central nervous system. So this just basically has given us a bit of information about where the start site are. They're confirmed in both a Down syndrome patient fibroblast line, as well as a medium spiny neuron, which is just a subpopulation of neurons in the brain. We've actually found a few more isoforms from further analysis. This does make a difference to some of our screening because this message here, this number means that there's 1,480 bases. So our genetic code is made up of bases, and obviously that's a large region that we can target, whereas in this section there's only 86. The good thing is there are two really attractive bowls in both of these transcripts that we're targeting. That seems to be a good area for us in our preliminary data so far. So we're hoping that it's not too much of an issue. So we initially screened our compounds in a neuronal-like cell line, a cancerous neuronal-like cell line, because they proliferate really nicely and they are a good cell line to study for early studies looking at neuronal conditions, because obviously neuronal tissue is quite hard to come by. We generally have to make them from skin cells or from stem cells. So what you're seeing here is a screen of 5 compounds at 2 concentrations. So the triangle, the one on the higher side is the higher concentration and the one on the lower side is the lower concentration. DYRK1A exists when you visualize it under sort of a chemiluminescent light in 2 forms. It's in its native form, so the way it's made. And then it also autophosphorylates itself. So what that means is it basically adds a phosphate group to the protein. You don't need to worry about what a phosphate does, but what that does is phosphorylation and dephosphorylation are how proteins turn on and turn off. So it's how DYRK1A is sort of involved in that process, and that's why there's so many large factors that are affected by DYRK1A, because there's so many downstream binding partners of the protein. So it exists in two forms, because when it does get that phosphate group, it gets slightly bigger. So these are separated on size, and really what I just want to draw your attention to is all of the treated, which are the samples in red. You can see that they're slightly darker. So the color of the blot and the intensity of the blot is relative to the amount of protein. And then these bands below here are what we call housekeeping genes. So it's to account for different loading. So if you only looked at one gene, you wouldn't know whether you added different amounts of that protein. So you need to use other proteins to ensure that it's been loaded evenly. So you can see that we are getting an increase up to twofold after incubating them for 72 hours, and then we collect them and extract the protein. We then repeated it a few times and got a bit of variability, and we just thought it might be due to Western blotting, which is the technique of taking a protein and visualizing it under specific fluorescence. It's a notoriously difficult experiment to do, and I think most scientists, if you ever ask them, it's probably one of their most hated things to do, because it's very unreliable, and as you can see, it's not always the cleanest. But again, what I'd just like you to appreciate from here would be showing you that these two are the control samples, and are treated again, appear to be darker, which then typically correlates to more protein on the membrane, and therefore more protein being expressed in the cell. So then we're able to obtain a patient's skin sample. So a fibroblast is just a type of skin cell. They're really good for studying genetics and using them as screening, because your compound's able to get into them quite nicely. So you can use them as an effective screening tool with someone who does actually have the syndrome, because it's all good and well showing good data in cell lines, but obviously we want to ultimately show that you can do this in patients as well. So from this, what I just wanted everybody to take away is this is an average of an unaffected individual or individuals, and the DYRK1A patient, you can see we normalized it. So if this is one-fold gene expression, so that's how much, if someone has two functioning copies of the gene, that's how much they'll express. So in a haploinsufficiency phenotype or disease, which is what DYRK1A syndrome is caused by, it's generally caused by about half the gene message being produced and then being made into a protein. So although this is not exactly half, you can see that the patient does have reduced DYRK1A expression when compared to various healthy controls. So when we screened our PMOs, which is a specific type of chemistry, we were really disappointed, because we actually didn't see an upregulation, and in fact, at times a little bit of downregulation. So here, these ones are slightly darker, so it means that there's slightly more protein. But anyway, when you use software to normalize this, we didn't get much of an increase. So we're bitterly disappointed, but that was one experiment. so we repeated it and then got mixed results as well. We got a varying amount of upregulation, some non-effect. But the more I work on upregulating AOs, this seems to be a trend that I encounter. I've chatted with a few people who published some of that data that those papers that were earlier, and they seem to be this really sweet spot of when our compound is effective at hiding the bowl of porridge, but sometimes it's too effective that basically the hand trying to reach the porridge is inhibited by getting the porridge. So that's why sometimes you can see a bit of decrease here. So it's just about managing dose and other factors related to the compounds. So for this reason, we looked at getting a different chemistry. I won't bore you with all the chemistry there, but basically what it just means is these molecules have a charge, so they're negatively charged. So that means that they can move through membranes and organelles a lot easier than the other compounds that have a neutral charge. So if you think about two magnets, if you put a positive and a negative, they'll come together, but anything that's neutrally charged, there'll be no attraction or repulsion. So the idea was to use a negatively charged compound. So the reason for this, and we also wanted to reduce the length because we wanted to reduce how tightly our compound binds onto the genetic information. This is sort of displayed as what we call a TM. We get better transfection efficiency, and it's a much quicker screening process. So we designed a whole more bunch of compounds, did the whole screen, and we got some really good hits, which are on a similar region to where we were before. But this is the start sites that I said were universal to both of those transcripts. So this made us really excited. We also found some information that not only does it potentially hide the porridge, but what it also does is it just keeps the porridge nice and warm as well. So it basically stabilizes the transcript. So again, what I'd just like you to take away from this is the bars going upwards are an increase in expression, and the bars going down are a decrease. So as I said, some compounds still do cause this decrease. And when you account for biological replicates, you can, you can see that the error bars, so this just represents the distribution of the results. So the wider they are, the more variable they are. And normally we like them to be really nice and narrow, and like what we like to say, tight error bars, like really nice and narrow. But you can see that the same compound's also giving us an increase in gene expression. So we're getting an increase in gene expression and then also getting an increase in protein expression. Then we did another N of 5 and got similar results. But we've subsequently repeated this and they weren't as favorable. So there was much larger distribution. As you can see from all the Western blots, they are changing over time. We're really struggling to get an antibody that works nicely for this protein because it's not a well-studied one, and in the context of diseased cells, there's less protein to be expressed, so we do have to load an extremely large amount. So beta tubulin does get out of its linearity, which is a bit of jargon, but really all I want you to take away from this is just that you can see here's our control. So we've normalized it to that, where we've designed a sequence that is a scramble of genetic code. It doesn't bind anywhere, and it shouldn't cause any effect. And then we've compared that to our actual DYRK1A targeting compounds, and you can see that we're getting roughly twofold, so double the amount of protein. And here probably sitting at around 1.5. So we've got a PhD student now starting this, which is really good because it's only been me, and I've sort of been just working with another research assistant. So it's good to get a full-time PhD student on this. So we still need to determine why the mechanistic action is unpredictable. It's got to do with a lot of things, but some more sort of mechanistic studies would probably help that. We're going to anneal or move where the sequence binds by a few bases, so really just moving it up and down a little bit. And we need to assess toxicity, because with the charged compounds comes the potential for toxicity. But if you design a proper oligo and you do the right experiments, you can negate all of those effects. So you just need to do a bit of toxicity and innate immune response. What we'll do is do a method known as RNA-seq. This looks at all of your genes in one go, and it can provide insights into sort of the genetic pathways that may lead to some of the physical manifestations that we do see. And then hopefully we're going to get some iPSCs, or we'll reprogram some cells so that we can confirm this in neuronal cells. So yeah, as I said, this has been taken on by Aidan. He did some of the later work. But yeah, thank you for the time, and happy to take any questions. And apologies, sorry, I might have missed that it was going to be a mixed audience. I really apologize.
No, this is perfect. Thank you so much, Craig. Really appreciate it. Does anyone have any questions? Yeah, go ahead.
Hello. Hello. Can you hear me?
Hey, Oliver.
Hey, how's it going?
Yeah, not too bad. How are you?
Great talk, Craig. Really appreciate your work. I just thought it'd be important just for the audience just to try to put in context, Craig's work, because I think the approach that he's taking it's very challenging, but it has incredible benefit. As he mentioned, one of the challenges in DYRK1A is it's a haploinsufficiency disorder. So you're only creating essentially half the amount of protein due to these mutations. Now, there's various ways that you can address those. You can address those mutations like an N of 1 example. So each person may have a different mutation. And so if you're going to do treatments, you may have to have a different treatment for every single person because of that type of mutation. The approach that Craig's using with these antisense oligonucleotides and sort of trying to upregulate gene expression, increase protein expression, that has the potential to benefit, any person or child or adult with DYRK1A, because it's not mutation specific. You're essentially just trying to upregulate the good gene. It's a very challenging approach. And Craig's done a tremendous job, just starting really from scratch and doing as much of the work as he has. So I just want to commend him for all the work, but also, since this is a mixed audience, just kind of wanted to help, put that in context of what he's doing and the approach that he's working on. So that I just wanted to mention that. Thank you.
Awesome, thank you. I was going to actually make a very similar comment, because I think it is important to know. I mean, we acknowledge almost all of our kiddos and our loved ones have an N of 1 mutation. I think for our son, he's the only one, and most are. So that's the question. If we're trying to upregulate, is it actually feasible to target every single mutation? It's not, right? It is, because actually that's what I do in my day job, but it's a lot of work. So that's the goal here, is to have one approach to potentially upregulate. I think the interesting challenge here is, like, how do you define how much would be beneficial? We have no idea. So I think that's one of the interesting things that you showed, Craig, is like going up to, 1.5 to 2-fold. It's possible that even just a tiny bit would help, right? We just don't know. So I think that's one of the really cool things about what he's doing. So again, thank you, Craig. We just really appreciate what you're doing, and also for staying up so late for us. So.
Yeah, no, that's all right. And I guess just on the second with that, to give a bit of context with, I guess, how much like is enough, I guess, is what everyone thinks is how much do you need. I understand it's going in the context of the Duchenne boys. Some of them were only expressing they were. This is a disease, a muscle disease where there's a loss of an anchoring protein. So like a ship's anchor, it anchors the muscles together. And our labs developed 3 drugs, but although they work really well, the boys only who received the drug only went from about 0.2%, relative to a healthy patient, to only about 1%, and that stopped the deterioration of their muscles. It didn't improve benefit, but it did stop deterioration. So hopefully, with a bit of the RNA-seq, you'll figure out the amount of upregulation you might need to balance out all the downstream effects.
Awesome. Well, thank you so much, Craig. We just really appreciate you, and you're welcome to stay, but you're also welcome to head off. So thank you so much.
All right. Thank you, everyone. Oh yeah, if you have any more questions, I don't mind.
Okay. Well, we can all also follow up with you. So thanks, Craig.
Thank you.
Thank you. It doesn't look like Amelie was able to join. Did you guys? No, I haven't seen anything. Okay. Okay. So next we'll move to Helen. Are you able to hear us and see if we can hear you?
Yeah.
Okay, great.
Hello. Good morning. Well,
Good
morning for me. I guess it's not morning for everybody.
No, well, thank you, Helen. So Helen's on the West Coast, and it's really nice to meet you. So Helen, if you don't mind just introducing yourself, and then we'll turn it to you to talk through some of your work on DYRK1A.
Okay, great. Let me go ahead and share so that we can make sure that works before I jump in. Oh, it's gonna, okay, I have to open system preferences. Sorry, we never used WebEx, so I just apparently have to give it some permissions. 1 second.
Mhm.
Oh,
Are you
I
using
have
a
to
Mac?
quit screen and come back. I'm on a Mac. I have to quit and come right
Yes.
back. I'll be right back.
I didn't want to. It's too boring. There's that one.
Oh, you tap our numbers. But, you know, my phone comes with the stylus, so it's a handle today.
Okay.
Oh.
Oh God.
Oh, she's gotta unconnect her other one. She's got 2 computers going. Yeah.
You have 2 computers going.
Or she has to mute the other one. And the other one get kicked from this. Like, can you guys kick the other one? Hang on, she just muted that. Let's see.
There.
Oh. Oh. Yeah.
That's what I just
Yeah,
said.
yeah, kick it out.
It's the Phantom.
We're happy to have 2 of you.
Yeah, maybe try just muting it because it doesn't look like.
Yeah, it seems fine.
There you go. That one? Yeah, no. Mhm.
How about?
Mommy, which one should I do?
And we know WebEx terms.
All right. We kicked out the imposter.
Oh, somebody's trying to join the meeting. It looks like it said not in the meeting, but if somebody. Okay.
share now. Yes.
There you go. Perfect. We can.
Yes.
Yep.
Yeah. Right, nothing like a little technical difficulty to start us off. Helen Willsey. I'm an assistant professor at UCSF.
Oh, wow.
Does
Oh, God.
frogs. You can see it. Yep.
Sorry, we have an echo.
No, we're not
here.
okay.
No.
Uh-oh.
Try to talk again.
Hello, can you hear me now?
Here we go.
We're okay?
I think so. Yeah, it's a little quieter, but there isn't an echo, so let's go for it.
Well, at least.
No, no.
Or I'm echoing myself. No, we're still there?
Yes, we're still here. We're still here. Can you hear us?
Yeah. Am I?
Yeah, no.
Hi.
Did you connect in computer and phone by any chance?
No, I'm going through my. Let's see. Craig, are you there? Does this problem have?
Here we go. Is this,
Yeah, I'm here.
and you're not echoing, right? This is a me problem.
I don't know. I can't hear myself echoing. I don't like the sound of my own voice. So I
Yeah,
think I'll hear
his
it.
connection is good.
Okay, something about me. All right. Yeah, I'll switch my audio to my phone. How about this?
I think we can hear you.
Yeah,
Now you can hear me?
Yeah, press the button.
yeah, no echo.
That's okay. Oh, fabulous. Okay, so we're learning a lot. Here we go.
There we go. Should turn the volume.
I just called in with my phone if that happens to
Yep.
anybody else.
Okay. It's
Okay.
perfect. Thank you.
We're good to go?
Yeah.
Yeah?
Yeah.
Okay, good. I run a research lab at UCSF. We use frogs to study a wide variety of neurodevelopmental disorder risk genes. One of those includes DYRK1A. So thank you for inviting me to talk and share our work with you today. Please interrupt me, ask questions, and I'll stick around for the discussion. Why do I use frogs, right? Why does that make sense? These frogs have a diploid genome that's very similar to humans. It means they have one copy from mom and one copy from dad. That's unlike zebrafish, where they actually have a duplicated genome that makes the genetics more tricky. And what's really cool about these frogs is that one mating pair can lay over 4,000 embryos in one day. So we have a lot of animals to work with and manipulate and see how they develop. And they develop after only 5 days into free swimming and behaving tadpoles with complex neurobiology and behaviors. And they're also really cost effective. So my animal costs are probably an order of magnitude lower than any other rodent model. Drug screening is also really nice. You just put the drugs in the water with the animal. So we have a vertebrate animal where we can do high throughput drug screening to understand and try to identify molecules that can reverse the effects of any genetic manipulations. But I think the coolest thing about frogs is something I'll show you in a second. But first, just to make sure that you realize there's actually a lot of similarities between human and frog DYRK1A. And so this is just an alignment of the protein sequence of the human version of DYRK1A and the frog version of DYRK1A. And any one of these letters that you see as red means that they're identical between the two species. So you can imagine that this kind of conservation of the protein sequence between these species bodes well for being able to manipulate it in frogs and learn something that'll be relevant to human biology. Okay, but the coolest thing about frogs is this. We can make half-mutant tadpoles. So we catch developing embryos right after that first cell division after fertilization, and we physically inject CRISPR-Cas9 genome editing reagents to be able to mutate whatever gene of interest that we want to study, in this case, DYRK1A, along with a red dye into that 2-cell stage embryo. And then we only wait 5 days, and then we have a free-swimming behaving tadpole where just half of the body, including half of the brain, which is right here, carries the mutation of interest. And so this is quick. It's $25 per gene, so it's high throughput and scalable. So we're doing this for many different genes. This is our typical afternoon where we've made thousands of half-mutant embryos. And this is right after the neural tube has closed, so still kind of early in brain development. This is a proof of principle we did early on where we mutated a gene required for eye pigmentation in just half of the animal on the right side. And you can see on that right side, the eye, which is right here, can no longer form pigment and it's albino, compared to the other side, which is our control, it's unmanipulated, where now you can see pigment. So from this kind of experiment, we can infer that the function of this gene is required for eye pigmentation. And so the idea is we can use this kind of half and half strategy to have an internal control to mutate these genes and say, what do they do during brain development? And the way we image them is largely through microscopy. And so we can label different kinds of cells with different colors. We can look at all of the neurons, only the neurons that are actively dividing, different kinds of neuron types like excitatory, inhibitory neurons, and even astrocytes and microglia. And we can look at them in the whole animal and the whole brain all at once. We can take out the brain, we can flip it around and look at it in 3 dimensions. We can segment it so we can compare the injected side compared to the uninjected side and start to understand, well, what happened when we mutated the gene on the injected side. Just to orient you to the brain structure, this is the olfactory epithelium, so kind of the nose part of the frog. This is the forebrain region and the midbrain and the hindbrain and the eye. There's the vagus nerve and the otic vesicle, which is like the ear. And this kind of highlights what's really nice about working in frogs, because you can look at many different organ systems and cell types all at once that have been properly patterned during vertebrate development. So we can look at the eyes, the heart, the gut, the muscles, the kidneys, and the ears. And today I'll tell you about a couple of these different organ structures that we've looked at so far with respect to DYRK1A. And so the first thing we wanted to do with DYRK1A was understand what are all the types of organs and tissues that DYRK1A is expressed in. So where is this gene on during development? And in frog, that's really easy because we can do something called RNA in situ hybridization. But the key point here is that wherever you see purple on these embryos is where DYRK1A is actively on. And so even in the very early embryo, before it's undergone a process called gastrulation, this is pretty early, DYRK1A is already on in the what's called the ectoderm. And then a little bit later, after the neural tube has closed, you can see it in this kind of punctate pattern all over its skin. You'll see it here, 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 here. That was a big clue to us. This is something called a ciliated epidermis. We'll get more back into what cilia are and why this is interesting to us, but that was a big clue at the beginning of this work. It's also expressed in the eye, and these cells here, which are called the neural crest, which really form the craniofacial structures. We'll come back to that again later. And then also in all the tissues that become the brain and the spinal cord. If you look a little bit later, again, you see it in some of those same tissues. You see it in so these cells here that become the craniofacial now also become the pharyngeal arches, which again contribute to the craniofacial structure, the ear, and really interesting, the pronephros, which becomes the kidney. And so I'm not going to talk about any of our kidney work today. That was in collaboration with Rachel Miller at UT Houston. But we have done some work about DYRK1A and its essential function for kidney and urogenital tract development. And so we can talk about that later if people want to get into that. Also expression in the heart. And then if you look a little bit later, you see it in this pattern right at the edge, the inner edge of the brain ventricle. Those are the cells that are actively dividing in the early brain. as well as in the eye and the developing ear. And if you look a little bit later, you can see it pretty broadly expressed throughout the entire brain. And so this told us a couple things. One is that this is not a brain-specific thing. You know, it's expressed in many different developing organs, and we think all of them are interesting to study. But the first thing we started studying was the role in the brain. And so what we did was that half and half analysis to knock out DYRK1A and produce loss of function variants in one half of the animal and compare it to the other side. And what we did when we did that, this is now a zoom in on the forebrain part of the brain. And hopefully what you can appreciate is on this side here, which has been mutated for DYRK1A, you see this dramatic reduction in this structure here, which is the telencephalon, or a portion of the forebrain, compared to the other contralateral or other side control that's been unmanipulated. So this in some ways recapitulates the idea of microcephaly, or a smaller brain size. I think in the last talk, y'all were talking a little bit of trying to rescue some of these variants with gene therapies. And one thing we've done in frogs is say, well, could we rescue this by adding back the human version of the gene? And so one thing we did was just overexpress the human version of the gene. It has this GFP tag, and just overexpress it on one side and compare it to the other. And what you may be able to appreciate is that actually you get a larger brain on the injected side when you just overexpress DYRK1A, right? And so to me, that really talks to the idea of the dosage of DYRK1A is really important to determine the number of neurons that end up in the brain. But excitingly, then when we combined them, so we knocked out the frog version of DYRK1A, but then added back the human version of DYRK1A, the human version of DYRK1A was able to step in and actually restore brain size to the proper level. So again, this gives some weight to the idea that the frog and the human proteins are able to compensate for each other and do similar things during vertebrate brain development. The next thing we wanted to do was say, well, why are these brains smaller? Like, what is going on here? What's the actual mechanism for why these brains are smaller? So we did what's called RNA sequencing. So we took all the RNAs out of that brain that's been mutated. And so we made a mutation right in the first exon of DYRK1A that we validated here. And then we looked at all of the genes that were being expressed and which ones were different between the animals where DYRK1A had been mutated versus the other ones. And we have quite a list of many different genes that are differentially on and off compared to the two. And one quick thing people often do is called a Go analysis. It's gene ontology. There's good things and bad things about Go, but sometimes it's nice to be able to use it to generate new hypotheses about what's going on. And this is really taking that list of genes that are differentially expressed and saying, are there any patterns among what these genes do? And the big take home for this analysis was that there's a lot about cell division. So it's about replication, cell division, DNA unwinding, cell cycle, cell cycle, cell cycle. And then what's really interesting to us was this idea of microtubule-based processes and microtubule cytoskeleton organization involved in mitosis, okay? So we looked at this and we said, okay, something is going on with cell division, and it looks like there might be something going on in microtubules. And because of this, and in combination with some work that the Jan lab at UCSF had done, showing that DYRK1A actually directly interacts with tubulin, which is the main component of microtubules, we looked at the mitotic spindle. So let me back up for a second. The mitotic spindle is the structure during cell division that organizes and actually splits the chromosomes equally into the two daughter cells. So you can kind of see up here the structure that kind of looks like this with the DNA, the chromosomes in the middle, and then it will actually split during cell division and cause the chromosomes to go into the two daughter cells. And so when we looked at where DYRK1A was on the structure, we could see it beautifully on the mitotic spindle, and then these little puncta along this tubulin-based structure with microtubules. So this led us to the hypothesis that DYRK1A is really important for cell division, and specifically its role could be on microtubules itself to help form the structure of a mitotic spindle. If that's true, we would predict a few things. So we would predict that when you had a loss of function DYRK1A, you would disrupt the cell cycle, so that would not be going properly. You'd get stuck at this stage where you're making your spindle because you wouldn't be able to properly separate the chromosomes. Then usually what happens is cell death if the cell can't properly split the chromosomes. So to test that, we used this chemical inhibitor of DYRK1A. It's a kinase inhibitor called Harmine, and we compared that to control animals. And what we saw was that, yes, we see disruptions and cell cycle progression. So these cells get stuck at this M phase. The M phase is the mitotic phase. It's right when that mitotic spindle is being formed and trying to split the chromosomes. So they're getting stuck during that phase, and then it's leading to pretty massive cell death throughout the brain during development. So this led us to the conclusion that, during brain development, DYRK1A is required for proper cell cycle progression, the actual dividing of chromosomes during mitosis, and then ultimately survival of these neurons through brain development. The other thing, and I'll come back to that idea that we saw DYRK1A was on in the skin in that punctate pattern. That was a big clue to us because that punctate pattern are ciliated cells. Okay, so what are cilia? Why do we hear about cilia? Cilia are these membrane projections from cells that are microtubule based. So just like the mitotic spindle, the pink here, these microtubule structures, cilia are another microtubule based structure that's really important for development. And so again, this is saying, where's DYRK1A relative to this microtubule based structure? Again, in pink, you have the microtubules, and in green, you have the DYRK1A protein. And we can see that, again, it's in this punctate pattern along microtubules as well as some other places. And this is really interesting to us because cilia are really important for a wide variety of roles during development. And if you have defects in cilia, it can lead to a wide range of multiple organ issues in a person, including some of the features that have been noted for DYRK1A haploinsufficiency. So certainly things like microcephaly, heart defects, kidney problems, respiratory problems, and on and on. So we thought that this was an interesting finding that not only is it on the microtubules of the mitotic spindle, but also on the microtubules of the cilia. And so we did some simple experiments where we inhibited DYRK1A. This is the control. There's all those beautiful cilia. This is a CRISPR. We also did this with a morpholino, which is like an antisense oligonucleotide, where we knock down the levels of DYRK1A, and you can see that you reduce the number of cilia that are being formed per cell. So this work that I've shown you so far is all published. And so what I want to go into next is some new work that's unpublished right now from a graduate student in the lab, kind of now zooming out and saying, all right, we have this understanding that DYRK1A is involved in cell division. We also know that DYRK1A is involved in these cilia. And so what does that mean on an organ level? Are there certain organs that can tell us additional things about how those deficits in mitosis and ciliogenesis lead to different organ structures. And so one of the things that's really interesting about gut development and microtubules specifically is not only do microtubules do the mitosis and the cilia, they're also really important for neuron migration. And so when the gut is developing, neurons from up, at the head side of the animal have to migrate all the way through the animal and then migrate into the gut to control the actual peristalsis of the gut. And I think one of the last times I was presenting to your organization, one of the things I learned from the families was the sort of struggles with gastrointestinal problems that many people face. And so we got interested in that and saying, can we learn anything about the gut and its relationship with DYRK1A? So this is the tadpole gut. These are the little neurons that kind of span the whole gut. What's nice about saying guts in frogs is that they're curved and coiled just like the human gut, and they have so many different cell types and different types of neurons. all together to actually control the movement and gut motility. These neurons, like I said, initiate really early in the embryo. And so what we wanted to do was look at that pool of progenitor cells that become those neurons of the gut and say, what happens when we lose DYRK1A? So let me just orient you a little bit. You can really just look at this one if you want. And so again, we've done this half and half thing with the frogs. So this side of the animal is your control, your unmanipulated side. And then this side, we've inhibited DYRK1A by CRISPR-Cas9 genome editing. And what you may be able to appreciate is there's this blob of cells here, right? This is a marker for SOX10 migratory neural crest. These are some of these cells become the neurons of the gut. And so you can see them really nicely in this pattern on the control side, and it's really reduced when you lose DYRK1A. And so these cells give rise to the neurons of the gut. They also give rise to some of the craniofacial structures. You could imagine that a loss of DYRK1A would lead to problems with gut neurons and also in forming craniofacial structures. We then wanted to look a little bit later at the actual neurons migrating into the gut. And here you can just compare these two really. And so now we flip the animal. So this side is the control side, and these are the gut neurons that have migrated from the brain and are now coming into the belly to actually coil around the gut. And you can see in the control situation, they've migrated quite a far way. They've made it over halfway through the animal. But if you turn this guy over to the side where we've inhibited DYRK1A, they've started to migrate, but they just haven't made it nearly as far. So they're having trouble actually migrating into the gut. And so this is getting a little bit into the details, but you could imagine that this migratory defect could also be because of that earlier defect in the actual pool of these cells. So we wanted to be able to disentangle these two. And so what we could do is we could take embryos where we let them develop unperturbed and form those neural crest cells just fine. And then what we could do was then add the DYRK1A inhibition and say, do we still see problems in migration? And so indeed we do. You can compare this one with this one. And so you can see in the top left, you see the neurons are all migrating perfectly fine into the gut. But even here now with the DYRK1A inhibitor, they're really not getting far at all and really having trouble migrating. So we think there's kind of two effects of DYRK1A going on here. One is just maintaining that initial pool of cells that eventually become the gut neurons and craniofacial structures. And then later on, they also have trouble migrating into the gut. So you could imagine then that if you have fewer cells, fewer neurons in the gut would have problems actually mobilizing with motility. The next thing we're really excited that we're working on now is we can actually feed these tadpoles fluorescent beads. And so here you can see these little beads that are inside the belly. So the gut is all right here. It's covered in this kind of aluminum foil type structure. But if you just look at the fluorescence of the beads, you can see it through the gut. And so now what we're doing is we're inhibiting DYRK1A in this system and then adding on 800 different FDA-approved compounds and looking for drugs that can actually increase the movement of these beads through the gut. And we think that those might be interesting compounds to think about for these gut motility problems. Okay, so what I hope you can take away from this today is that the human and frog versions of DYRK1A are very similar, so it makes sense to be studying DYRK1A in frog. We need to take advantage of all the advantages of frog biology and experimental feasibility to make some new insights about the function of the human version. DYRK1A is expressed in many different developing organs, spanning the kidney, the brain, the heart. So it makes sense that patients with loss of function variants might have multiple different organs involved. And then really what we're honing in on is that DYRK1A is really important for these structures, microtubules. These are kind of highways of the cell. They're doing a lot of different things. They're important for cell division. They're also important for these cilia structures. They're also important for neurons to be able to migrate. And so we specifically now looked at gut neuron development and see that there are migration problems and neuron number problems without DYRK1A. So with that, I'd like to acknowledge my laboratory at UCSF and our Xenopus resources and our funding. So thank you for your attention. I'm happy to take any questions, or if we want to save them for the discussion, that's fine with me too. Thank you.
Awesome. Thank you so much, Helen. Really appreciate that. Anybody have any questions?
Hi. So I just had a question. Have you ever done a study where you've mutated the full frog with DYRK1A and then had like a typical frog, and to compare the two, like if they had both sides of the brain that were affected with DYRK1A versus just one side?
Yeah, it's a great question. So we're making the mutant germline line now with the Xenopus resource. It's like a stock center. They will basically make it for us. So that's in progress right now, making those frogs. What I can say that we've done in terms of like both sides is when we use that chemical inhibitor to inhibit the kinase activity of DYRK1A, that happens on the whole animal. And so we have looked at those, and that does cause a reduction in the total brain size, both sides, when we add that chemical inhibitor. So yes, we're doing it. It's coming. Not quite there yet.
Thanks.
Yeah.
It's a great talk, Helen. This is Oliver Glass. I just had a question in terms of, you showed some great data early on in terms of rescuing sort of the phenotype with DYRK1A in the brain. Have you all done any sort of more work with that in terms of just, I think we're all really interested to just better understand the sort of like window of opportunity of treating or rescuing DYRK1A. It's like if you already have the brain development within the tadpole that has loss of DYRK1A, like are there ways where you can kind of rescue it? Have you done any sort of those like time course assays or experiments?
Yeah, that's a great question. Certainly really relevant. So that experiment that I showed where we were rescuing with the human version, that was from very early in development, right? And so I think that doesn't get at your question of a developmental window, or after brain development has already happened, then can you reverse things? We haven't done any more work on that, but in listening to your question, I did have the idea that what we could do is add the drug that inhibits DYRK1A throughout brain development, right, and get this phenotype of the smaller brain and the cell death and things like that. And then we could just wash the drug out, right, and then see what happens. How does it recover compared to the controls, right? So we haven't done that, and it's an easy experiment. So I might actually get one of my summer students to do it. So stay tuned. That'd be easy. Yeah, it's a great idea. Genetically, it's a little hard to turn things on and off in frogs, right? Like I think a mouse lab could do it with some Cree-lock systems and be able to mutate it and then add it back later, or something with ASOs. And that's a little more tricky in frogs, but I think we could take advantage of the small molecule inhibitor and use that temporally to see, because then you've added back the perfect amount of DYRK1A because you never really lost the gene. So we'll try that. I'll let you know.
And then just sort of follow-up question to that is, I know you're using the CRISPR-Cas9 system to sort of completely, abrogate DYRK1A sort of in half the frog. If you've looked at using just like an antisense or an RNA sort of based mechanism to just sort of knock it down instead of like completely losing it, so you can kind of mimic that heterozygous or haploinsufficiency phenotype, just to see if what that phenotype is like compared to complete loss.
Yeah, totally. So some of the stuff I didn't show you, but is in those papers, we used what we call a morpholino, but what I think now is more typically called an antisense oligonucleotide, to knock down DYRK1A. So we have done those experiments. We see the same thing, though we were adding quite a bit of it, right? And so I think we could dial that back to try to get 50% knockdown. We have not done that. We've done different concentrations of that drug, and you can kind of see a dose response as you lose more and more of it. But I think the real way to do that we're going to work on is with those heterozygous mutant frogs, right? That'll be the best model to be able to look at dosage effects of DYRK1A loss. So it's a great point, and it's something that's on our minds. I think in our case, we wanted to see what the full, I'm a geneticist, so we always want to see what the full loss of function is, so that we can know the full extent of the phenotype and then dial it back. So I definitely think we're in the dial it back now zone of our analysis.
Yeah, thank you.
Hi there. I just had one quick question. I don't know if you could expand upon the kidney. You mentioned that you were seeing that it is important in kidney development. I would just like to know a little bit more about what you do know up to this point, and if it's something we should be keeping our eye on. Does it have anything to do at all with potty training? Is there some sort of signal that's not being sent to the brain to indicate earlier on? Maybe, I don't know. This is a total somebody who's a total lay person, but just what you could tell us about the kidney and what you know. Thank you.
Absolutely. Yeah, so I think this is a really important area for a lot of these epileptic encephalopathies that hasn't really been explored much. And so there's this, I'm an author on this paper, but it was really led by Rachel Miller at UT Houston. So I can send that paper out if people want to learn more about this. But what they did was they actually had a cohort of, I think, 19 patients with DYRK1A haploinsufficiency, and a striking percentage of those, really high, had congenital anomalies of the kidney and or urogenital tract, right? So that structure is kind of combined in terms of its clinical presentation, from my understanding. Again, I study frogs. I'm not a clinician. But in that work, that really suggests that there's going to be a high frequency of kidney and sort of urinary and urogenital tract anomalies in these kids. And I think a lot of times, if somebody's not looking for that clinically, it gets missed. And so that would certainly be something to talk to a physician about if there's any suspicion that something might be going on in those organ systems. And so in that paper, what Rachel did was inhibit DYRK1A during development, and you saw these very large kidney sort of development and organization problems when you would lost DYRK1A during development. And she was able to rescue that by adding back DYRK1A. And then what I think was really cool about that paper is that she added back a patient variant version of that, and that couldn't rescue the kidney problem. And so for me, that's really strong evidence that DYRK1A is doing something really important in kidney and urogenital tract development that I think is worthwhile to look at clinically, though again, I'm not a physician. And, nobody's followed up on this yet. I'm thinking about doing it a little bit in rats, actually, but for the kidney to develop, it needs those cilia. The cilia are really important for kidney development and actually to sense the fluid flow through the kidney. And if that's disrupted, you get pretty serious kidney problems. And so my idea about what DYRK1A is doing is through that cilia mechanism is disrupting kidney development. And so that's my wild speculation about why there could be kidney problems. But I'll send that paper out so that you all can take a look. But yeah, a huge proportion of patients with DYRK1A haploinsufficiency had kidney issues. Did that answer it?
Great. Thank you so much. Sorry, I had to step out for a second. My son is running around. So thank you, Helen. We just really, really appreciate you so much, and really excited to hear all the work you've done. I just personally learned so much. Honestly, I didn't know most of what you shared, so thank you for that. So Amelie, we see you joined, but also acknowledge, I think you needed to leave early. So just wanted to turn over. Do you have time for a short presentation or?
Hello, everybody. Can you hear me?
Oh, we can. Yes.
Okay, perfect. I'm so sorry because at the hospital, like everything was totally blocked. And so I had to come home to be connected. So yes, for sure, I can arrive later at the school. It's not a problem. I managed to. So maybe I can. So I'm sorry I have missed Craig's presentation and a part of Helen's one.
That's all right, don't worry.
Oh, did we lose her? Quick question while we're waiting for Amelie to get back. Is there any way that Craig and Helen could share your PowerPoint presentations with us?
Yeah, absolutely.
Yeah, sure.
Who should I send it to?
Who should they send it to?
Yeah, you can send it to, and whoever you have contact with is fine, but, or you can do the admin@DYRK1A.org.
All right.
Great.
And the presentation is being recorded and it's going to be in our DYRK1A channel at YouTube.
Okay, I'm sorry, I'm back because I don't know why it froze. I couldn't hear anything. Can you hear me again?
Yes.
Yes.
Okay, I will try to share again. I hope it will not disconnect me. Is it okay? Can you still hear me and see something?
We can still see you. Yeah, there.
Yes.
Okay, got it.
Okay, perfect. So sorry. I don't know if it's France, so we have like a lot of connection problem. So hello everybody. So I will maybe present myself first. So I'm Amelie Piton. I'm working in Strasbourg at this institute, the EGBMC. So it's the east of France, and I'm working in the department Translational Medicine and Neurogenetics. And actually, I will present you the work we are doing in the team. But there is another team also from the same department working on DYRK1A, the team of Yanero, and they are working with rat and mice model of DYRK1A for a triplication of DYRK1A, but also an haploinsufficiency of DYRK1A. And so we are collaborating closely together. But I will not present his result today, but they are doing a lot of work also on DYRK1A and on DYRK1A syndrome. So in the team, we are working more on genetics of neurodevelopmental disorder, but so we are also trying to understand the pathophysiological mechanism involved, for some of the genes, so you know now that there are now more than 1,500 genes involved in neurodevelopmental disorders, so we cannot study all of them, but we are working particularly on DYRK1A because of the history. In the first screen we did, we identified several mutations in DYRK1A in different individuals, and that's why we began to be interested by this gene. And so in the lab, we are trying to develop some novel tools for genetic diagnosis, but also to identify novel genes. But we collaborate a lot with clinical geneticists and child neurologists to understand a bit better what is a genotype-phenotype correlation, and to do more clinical characterization for some of the neurodevelopmental disorders. Try also to understand the pathophysiological mechanisms to see if we could develop at one point some therapeutic approach. So for DYRK1A, the idea is to establish a list of the mutation repertoire, to try to work a bit on the mutation effect. But we are also trying to understand the consequences of DYRK1A loss of function using cellular model. I will present you that a bit. And we try to identify also some biomarkers to improve the clinical characterization. And I will present you one example, for instance, of a study we did to characterize the neurocognitive and neurobehavior profile in DYRK1A syndrome. So first, the clinical characterization. So we do that with some clinical colleagues, and we do that using three different ways. So the first way was to collect clinical files from clinical geneticists in France and also in Europe, and to add all this clinical information, to collect this clinical information via this colleague. But we also use the website that was developed by one of my colleagues, Strasbourg, which is called Genida, which is a website where parents can register, and it's dedicated for all the monogenic form of neurodevelopmental disorders. And you can enter the name of the gene, and you can answer to a lot of different questions about the clinical manifestation of the individual. And so we the data from Genida for DYRK1A syndrome, and you can see here that for now 33 we have maybe a little bit more now, so around 30 families that answer the questionnaire, and so we can collect some clinical information directly from the families. And finally, we also perform some parental interview with French families. So it was my clinical geneticist colleague who had also an experience in child psychiatry who performed this interview, and the idea was to measure the adaptive capacity using different scales. So he used the Vineland scale, but also to measure the presence of the diagnosis of autism spectrum disorder, and to characterize a bit more. So what is the neurocognitive and neurobehavior profile in these children with DYRK1A mutation? So first, with the clinical file, we collect all the frequent clinical signs that were present by the children or the adults with DYRK1A mutation. And you can see here that finally, the most frequent signs are intellectual disability with impaired language, microcephaly that are found also in all the patients, feeding difficulties, epilepsy in half of the patient, anxiety, autistic traits, and etc. And so we collecting all this data, we were able to identify also additional signs that were not reported so far. For instance, we have like some of the children from the cohort who present some really atopic skin. So you can see here a picture of one of these children. We present really a severe dermatitis, and so it was really possible because of the collection of all this clinical file. But we also use this clinical file to establish a clinical score for DYRK1A syndrome, and you can see here. So we established this score on an initial cohort. So it's a score on 20 points, and so based on these recurrent features, but also based on the facial traits of the children. And so all the children with DYRK1A mutation present a score above 13, and we replicate that in an additional cohort of individuals with DYRK1A syndrome, and we just took some other neurodevelopmental disorders with monogenic origin, but affecting another gene than DYRK1A, and we were able to show that we don't have the same, we have like really low score for this neurodevelopmental disorder. So it means that the score was kind of specific. And why we developed that? Because for us, it was useful first to suspect DYRK1A syndrome. So it was more important before the development of next generation sequencing. Now that we can have access to exome sequencing or genome sequencing, it's maybe less important to really suspect it clinically. But it really helps us now to interpret variant of uncertain significance. So it means that when you do some exome or genome sequencing, sometimes you will find a variant in DYRK1A, but you are not sure that this variant can really affect DYRK1A functioning, and you are not sure that this variant is really involved in the disease of the individual you are looking at. And so the idea of the clinical score is to be really sure that the individual carrying this variant in DYRK1A is really like recapitulate all the clinical signs associated with DYRK1A syndrome, and it can help to interpret this variant of uncertain significance. So with Benjamin Durand, so the clinical geneticist, so he performed a parental interview to see what is the importance of autism spectrum disorder, because we know that autism spectrum disorder is really highly.
Just one question: are
yes,
you moving the slides?
Yes, you are not. You are not. Okay.
We are still in the clinical characterization slide.
Okay, so I will stop and do again. I think it's like a definitively not working. So you are stuck in which slide? Are you seeing the slide with the DYRK1A, the clinical score?
Okay.
You are seeing it?
Yeah, we have with the two pictures now. Yes.
Okay. Okay, so I'm sorry, I will move to the next one, but yeah, I will, okay. I hope it will go well now. So Benjamin Durand, so he performed some interview with parents to ask if the children have had an autism spectrum disorder diagnosis, and you can see that in more than 30% of the case, it was the case. So autism was clearly established. He also passed a Vineland scale to evaluate the adaptive behavior of children with DYRK1A syndrome. And he compared, for instance, with another neurodevelopmental disorder from genetic origin caused by mutation in KMT2A. And what you can see is like here is a communication score, and you can see that it's really more homogeneous for children with DYRK1A syndrome than for the children with mutation in KMT2A. And this communication score is really impaired, and it's really something that come out from the parental testimony. So in Genida, in the website where parents enter directly the clinical information for their children, there is one question which is, what is the major problem of quality of life for your children and from your family? And you can see here a compilation of all the answers, and there is really the communication that come first for a lot of the family. And it's true that when you look at the Genida data, you can see that more than 50% of the individual with a mutation in DYRK1A, they have like no language. Some have words only or can speak, but they are difficult to understand. And finally, there is only a small portion of the individuals who can do some correct sentences. And so here is a different communication way the individual with DYRK1A syndrome used. And what was really also impressive is that there is a question about do your child understand what is being said? And you can see here that in most of the case, the answer is yes. So there is really a big difference between what is understood by the children and what he can communicate. And it's really something that comes from all the different testimonies from the parents. You can see here, she understands a lot of more than she can express. She understands the majority of what she is told, but struggles to express herself. And it's really something that comes from different families. So it was so that was a part of the clinical characterization, but in the lab we are more doing directly in the lab a characterization of the effect of the different mutation identified in DYRK1A. So you can see here a view of the protein. the kinase protein with the kinase domain here in red. And there is an important amino acid here, which is the tyrosine in position 321, because DYRK1A will autophosphorylate, so activate itself on this amino acid position to be able to exert its kinase function. And so all the different mutations, so it includes some gene deletion, truncating variant in different parts of the protein, and also missense variant, so only one amino acid change, usually occurring in the kinase domain, but we have also other amino acid change occurring in the rest of the protein, and we are not always sure that they could be really pathogenic, so we are not sure they are affecting protein function. And that's why we develop a functional test. So you can see here the different missense variants we test. A lot of them are located in the kinase domain, but some others are located at the end of the kinase domain. And we include here a variant that will lead to a stop codon, so that will so the protein will miss one part here. Because the protein still have its kinase domain, we wanted to see if this variant could be pathogenic or not. So to test that, we include also different variants here in the coding sequence of DYRK1A, and we transfect cells. And we analyze in these cells the ability of all the protein with a different mutation to autophosphorylate, to activate themselves. And you can see here an example of a result, for instance. So you can see here the protein DYRK1A and the protein which is phosphorylated on this position. And we can see that for some of the variants, we had really a loss of this activity of autophosphorylation of DYRK1A. And here it's like the summary of the effect of the different variants. You can see that most of the variants located in the kinase domain here will totally abolish the ability of DYRK1A to phosphorylate itself, except one here that was finally classified as non-pathogenic. But for the other one, we could consider, we could finally confirm that they are pathogenic and they are involved in the clinical feature present by the individual, and so they have a DYRK1A syndrome. But what you can see here is that the missense variant located outside of the kinase domain, they seem to not affect the autophosphorylation ability of DYRK1A. What was interesting, it was this variant, which caused like a stop codon here at the end of the protein. We couldn't detect it because so here you can see the localization of the mutant protein in this cell, and you can see different points. It means that the protein with the mutation will lead to some aggregates. And I think it's because the protein will aggregate that it will lose the function in the rest of the cells, and so that's why it was pathogenic. And it was interesting because this mutation had some amino acid, abnormal amino acid, that it will lead to aggregation here in the cell. And if you replace here the mutation by a stop mutation, so really the protein stopped directly here without adding some supplementary amino acid, in this case, you will have no effect on DYRK1A localization and no effect on the activity of DYRK1A. It means that if you identify a variant here in this region, it's really difficult to really directly conclude that it's pathogenic and it's involved in DYRK1A syndrome. And so we need to be really, really careful when we have some variants outside of the kinase domain, and we need to perform some functional analysis to be really sure that they are causing DYRK1A syndrome. And in addition, we identify, so in the lab, we are trying to collect different variants located here outside of the kinase domain, and we identify several variants that occur de novo in the individual. And the individual really doesn't present at all the DYRK1A syndrome. So they have no microcephaly, so they maybe sometimes they have like more, an increased head size. So we think that maybe variants in this localization of the protein can lead to another neurodevelopmental disorder, which is kind of different of DYRK1A syndrome, but we are working on that right now. But coming back to the DYRK1A syndrome, which is really caused by mutations that lead to a loss of DYRK1A activity, we wanted to see, because we know that DYRK1A can have a nuclear activity and can regulate some proteins that are involved in chromatin remodeling. And so we wanted to see, we did that with a Canadian collaborator, Rosanna Wexberg. we wanted to see if in the blood of the patient, in the DNA blood, we can see a specific signature, which is a methylation signature that can be different between the patient and control individuals. And it was the case. So you can see here the analysis we performed shows that for 400 positions on the genome, we have a difference in the methylation status between patients with DYRK1A mutation, pathogenic DYRK1A mutation, and control individual. And so it's represented here, but you can also represent here this result in a multiple component analysis. And you can see here that you have the control here, and you have the individual with DYRK1A syndrome. And so we use this kind of biomarker. So it's a blood biomarker of the DYRK1A syndrome. And we also use this information to classify the variant of uncertain significance. And what you can see here in yellow, here is all the patient carrying a variant for which we found a functional effect on DYRK1A function. And here, all the yellow here, they were found in some individual for which, when we did the functional test, we had no effect on DYRK1A function. So it was really a perfect correlation between the result on the activity of DYRK1A and this biomarker in blood, which is the DNA methylation status. Oh, I will skip that. So in the lab, we are working to understand a bit more the effect of DYRK1A mutation and DYRK1A haploinsufficiency. We are working with neural stem cells, which are cortical neural progenitors. So these cells are able to proliferate and to self-renew, but they can also be differentiated into cortical neurons. We characterize in these cells the interactome of DYRK1A. And we found different interactors, and a lot of these interactors are involved in regulation of cell cycles, which really are really finally what we could expect for DYRK1A. And we also identified some novel interactors that we are trying to characterize now. We also did the first inactivation of DYRK1A. So for that, we use some scRNA to inactivate like DYRK1A expression. And so it leads to an inactivation of 50% of DYRK1A expression. So it can mimic a bit the haploinsufficiency, even if we know that this inactivation can be heterogeneous between the different cells. And so what we were able to see when we inactivate DYRK1A, we have here we look at the proliferation ability of the cell. And it's what is in blue here. You can see a total decrease of cell proliferation when you increase you inactivate DYRK1A. We also wanted to see what are the genes that are deregulated when DYRK1A is inactivated, and we perform RNA sequencing. And you have here, we identify a lot of genes that are down-regulated when DYRK1A is inactivated, and only few that are up-regulated. And I will not have the time to detail all of them, but some were particularly interesting because they are involved in regulation of cell state, cell cycle, and other things. And so just to, so what we are doing now, so because the problem of this inactivation, that it was a transient and heterogeneous inactivation, we are not able to really control which cell will have DYRK1A totally inactivate and the other one, it will not be so inactivated. And also we cannot, it's like a transient, we cannot really continue the inactivation in time. And so to overcome this problem, we generate some human-induced pluripotent stem cells, having a mutation. So we create some mutation in DYRK1A, and so by CRISPR-Cas9, and you can see here the two mutations we introduce in two different IPS lines. So they are really premature mutation before the kinase domain, and so it will really lead to a loss of function of the mutant allele, and so to haploinsufficiency of DYRK1A. So we generate this cell and we differentiate them in human neural stem cell to be able to look at the proliferation. So it's something that we are doing now in the lab, and also to look at gene expression by RNA sequencing. But we can also differentiate this neural stem cell into mature cortical neurons. You can see here an example of the differentiation. And what we are doing now in the lab, we are trying to characterize what's the effect of mutation in DYRK1A on the cell differentiation, on the ability of the cells to form synapse, but also on the neuronal activity, so the electrical activity of the neurons that are differentiated here. And we are also generating some whole brain organoid with DYRK1A mutation compared to wild type to see if it can affect the size of this mini in vitro brain, but also to see if it also affects the cell lineage, the different cell lineage we can obtain in this differentiation. And the idea at the end, so we have some fibroblasts of different individuals with DYRK1A syndrome. And so some of them, we already reprogram them into iPS cells. And the other, we will do it. And the idea is to confirm the result we will have identified in this cells that carry a mutation induced by CRISPR-Cas9 in patient cells directly. And so the idea is really to characterize all these mutant cells and organoids to find some readouts that could be useful to understand the consequences of DYRK1A haploinsufficiency, how it affects brain development, but also to have some readout to maybe do some job screening afterwards. So just in conclusion, so we are trying to continue a bit the clinical characterization with the different colleagues we have in Strasbourg and in France. And so we characterize the language alteration, but we are also doing some other clinical characterization, for instance, the type of epilepsy or the MRI anomalies, etc. So for the molecular diagnosis, we are trying to understand the effect of the mutations that are located really at the end of the protein to see what they are really doing. And so we are continuing the investigation in these cells for which we have introduced a loss of function variant in DYRK1A, so the neural stem cells, the differentiate neuron and organoid. And also we have another project that we will begin soon that is trying to understand a bit more about DYRK1A regulation and how we can maybe increase a bit DYRK1A protein level to see if we can restore some phenotype we will have in these cells. But it's something that it will be, I think, maybe touchy because we need not too much DYRK1A protein, and so we will have to find like really a fine regulation of DYRK1A protein. So here it's the team in Strasbourg, and so we have like collaboration with a lot of clinicians, so clinical geneticists, child neurologists, and child psychiatrists that really are really helpful to characterize more the DYRK1A syndrome at the clinical level. And we also have, so I was talking about Yanero's team, so we collaborate a lot with them, and so they have some rodent model for DYRK1A syndrome. So they have rodent model which have like one copy of DYRK1A mutated, and also they try to inactivate DYRK1A in some of the cells, for instance in glutamatergic neuron or GABAergic neuron to try to understand a bit what are the contribution, what are the pathophysiological mechanisms involved in DYRK1A using this rodent model. So thanks a lot for that. I will just finish by a picture of the DYRK1A meeting that was in Saint-Malo at the end of last year. And you can see, so we finished the scientific meeting with a family day. So it was in French, so it was more family from France. And you can see here all the different families that were there. And we tried to do a summary of what was found, what was presented during this scientific meeting for the family. And it was really a lovely day. Thank you. And I can take questions if you have some.
Thank you so much. Any questions?
I guess we'll just ask if she wasn't online when we asked if they would share their PowerPoint.
Are you willing to share your slides, Amelie?
Yes, of course.
Okay, that would be great. This is Sarah. I had one question. Thank you for that great presentation. I'm really curious about the dosage effect, and I know Craig talked about this a little bit too, about the Goldilocks issue that we have, not too much and not too little. And so I was wondering if you think in the context of treatments for DYRK1A, if we're trying to increase, like, are there biomarkers, like you talk about the autophosphorylation, or other really straightforward ways that we will be able to measure DYRK1A if we're trying to treat? Because obviously we wouldn't, you need to be able to assess that. And I think I've heard even recently FDA is asking for that, and a lot of these genetic therapies is like, how are we measuring the target? So I was just wondering if you have thoughts on that.
Yeah, I think it's really a difficult question. I hope that, for instance, the biomarker we find, the DNA methylation biomarker could be helpful, because we know that in patients with trisomy 21, so 3 copies of DYRK1A, but also 3 copies of other genes, they will have a kind of methylation signature that could be kind of mirror of what we have with DYRK1A syndrome. So maybe with this kind of biomarker, we can follow and see if we are like not increasing too much, or we could maybe see something there. In our cellular model, it's something that we would like also to investigate, to see if we can find a way to increase a bit DYRK1A level. What will happen to, for instance, gene expression? So we will have all the other readouts that we identify in the cell. So we will try to follow a bit that. But I think it's really a tricky question, and also we don't know exactly what is the really. So we know that the dosage is important. So we know that 2 copies is the best. But what is the I don't know how to say. What is the range we have? We know that only 1 copy is not good, 3 copies is not good, but in between, we don't know exactly what is really this right window. And so maybe I hope that by working on some different variants that could affect DYRK1A function, but maybe not totally, maybe just partially, we will have maybe more clue about what is really the right window dosage. I don't know if I'm clear, I'm sorry.
No, that's really helpful. I mean, it sounds like it's just difficult, right? There's not a straightforward answer yet.
And it's just a dosage. After, for sure, there is the window in development. So at what point, like too much DYRK1A, it's maybe deleterious at one particular step of the development, and having like less DYRK1A will be maybe deleterious, and another step, it's not so clear. So, or maybe it's the same step, or maybe the same, I don't. We have really a lot of work to do to understand that.
Yeah, no, that's great. Thank you. Any other questions? Well, if not, I think we just want to close by saying thank you to each of you, and also Lucas Branicki is also a member of our MSAB. Lucas, thank you for joining. We just appreciate your support. And if there aren't any other questions, then we'll go ahead and close the session. Thank you all so much.
Yes, thank you.
Thank you.
Thank you. So sorry again for all the connection problem.
No problem at all. Thank you.